Okay, good morning. It is my great pleasure to introduce Nuno Bandeira, a faculty with a joint appointment in computer science and in pharmacy, right? In University of California, San Diego. Nuno braved great travel adversity to come here. He landed, I think, 2 hours ago or something like that. We're very grateful for Nuno to be here. And the reason why we wanted to make sure that Nuno had a chance to present is that a lot of time when we work with the type of data that we discussed for the last 2 weeks, we want to do more than just analyze our own data, but we also want to rely on the data which are available elsewhere, where people have worked on a similar problem or maybe using similar technology, and we want to learn from that in terms of how to do the analysis, but also in terms of the actual biological discoveries. And so Nuno has done a lot of work in this area, and we really are very happy to have him here.
Thanks, Olga. All right, this seems all right. Can everyone hear me? All right, good morning, everyone. So as Olga mentioned, it was a bit of a tricky way to get here, but I'm glad that I was able to make it here and to talk to you a little bit about the sorts of things that we've been doing at MassIVE, and as Olga mentioned, how we look at making public data work for you. If it doesn't give anything back to the community, then we don't really see a purpose in doing it. So I'm going to give you some thoughts on the things that we have done, some of the resources that are available that you guys can use already, and also like it to be sort of a lecture style type of thing, not just a talk, saving questions towards the end. Ask questions at any time, or if you have suggestions, comments, thoughts, I'd be glad to hear them. Because as I said, it's really the intention is that the resources be something. This is having a little bit of feedback. I don't know. Maybe we can turn the volume. Let me get. Oh, it's right there. Okay. All right. So is that a little too low? I don't know. Can you guys still hear me? Can you hear okay? All right. All right. We'll adjust. Thank you. Okay. All right. So community data. What are we talking about? So you probably heard of the Proteome Exchange Consortium. Show of hands. Who's heard of the Proteome Exchange Consortium? About half, I guess. Maybe a little bit more. Okay. Thanks. So the Proteome Exchange Consortium is a group of repositories worldwide where if you want to deposit proteomics mass spectrometry data to meet the publication requirements of the journals, they usually require that you make your deposition in one of these repositories. The reason why there's been an effort to recommend that the depositions be made in one of these repositories is because there's a strong commitment to having open formats and to making the data as reusable as possible so that you don't just deposit the data somewhere and then you cannot do anything with it after that point. So there are a few repositories that are part of this consortium. You have PRIDE in the UK, MassIVE here in the US, and then PASL also in the US, excuse me, but more focused on targeted proteomics data. Then JPOST in Japan and IPROX was not in the consortium at the time, but now is in China. And this just gives a little bit of a distribution of where the data was coming from back in 2016. At the time, it's about 2 years, actually 3 years now. At the time, it was about 4,500 data sets that were available. There's about 8,000 public data sets now, and over 10,000, including data sets that have been submitted but are not yet public. So the papers are in review, so the data is not yet public. But out of these several repositories, PRIDE and MassIVE are the ones that are most dedicated to your typical type of mass spectrometry data, just the data-dependent types of acquisition. And altogether, we pretty much store the majority of the volume of the data, because that just tends to be the largest data sets that you have. So at MassIVE, which actually stands for Mass Spectrometry Interactive Virtual Environment. This is kind of the picture as it stands around today. So there's about 9,000 public data sets that altogether contain over 2 billion spectra, close to 200 terabytes of data. And it's close to 4 million files. And the first thing that might come to mind when you look at that is, okay, what am I going to do with 2 billion spectra? Right? And that is the question that we want to ask and want to address is, how can you make use of this information? As it is right now, we've used this to identify over 10 million different modified peptide variants, and I'm going to tell you a little bit as to how it is that that came about. But maybe the first step that I'd like to cover is just to give you an idea of what would it look like if you want to submit your data. You've been discussing here how to analyze it, you acquire data, you work with data. How is the submission process? How does that work? So for this first part, I'm going to give a bit of an overview of how the process works. And then in a hands-on session that we have afterwards, we're actually going to go and do submission of some quantification data for data sets, okay, which you would essentially build on the things that you've already been doing. And it's what you would do when you want to submit it and share that data. Any questions so far before I get into a bit more detail? Please go ahead.
Like if we are based in Europe, we can basically still, we have a free choice when to submit our data to. Right?
Absolutely.
Right, or MSGF.
Absolutely. All of these repositories, well, okay, I'm not sure I can speak for JPost and iProx, but definitely Pride and Massive will accept depositions from anywhere. And generally what we tend to see more is that because of the speed of internet connections, depositions tend to be made at the repository that's geographically closest, just because there's that intrinsic intuition that if you're geographically closer, you'll be internetally closer. And that's not always the case, but that's usually what tends to happen. But there's no policy, there's no blocking of depositions coming from anywhere. Thanks. All right, so how do things start at Massive? So if you go to Massive, URL up there, you just click register. There's a couple of fields that you fill in, and you then get an account. We'll actually go through this process this afternoon. The next thing you would do is just upload some files. And this is just one particular client that you can use for that purpose. When SCP, you just use FTP, go to Massive, use the account name that was created from there, and then you just drag and drop your files. Okay, and once your files are in your account in the system, then you can create a data set. So the way that things usually work is that as you're doing your research, your files get uploaded into the system, but everything is still private. The files are just there, stored in your account. And then when you're ready to create a data set with that, because you're done with the research project and you want to prepare your paper and you need to get an identifier to send to the journal, then that's when you get into this process where the files that you generated from your research can go in any of these different categories. Generally speaking, there are two types of submissions that you can make. There are partial submissions, where it's required that you submit the raw files from your experiment, as well as the search results from your search engine. But this can be in whatever format your search engine gives you search results. It doesn't have to be an open format. So at least there's something in a data set. However, it can't be reused easily because these tools typically use file formats that are not open, and it's not clear how to actually extract the information from those to make it reusable. It's much more preferred to have complete submissions. Okay, and what happens in complete submissions is that not just we get the identification results in standard formats, such as MZTab or MZIdentML, which you can then open with other software and you can read because it's an open format. But we also have the spectrum data in open format. Okay? The MZML is really the one that's most supported, but we also accept MZXML for that purpose in older format. Okay? So as it stands today, and I'll talk about that in a little bit later, but most of the submissions have been partial, but we really want to move in this direction, where complete submissions are really what makes the data more usable and reusable, which is really what we'd like to do for all of you to be able to then benefit from the data rather than it just sitting there. Okay. So a couple more things that go in a data set. There are some metadata terms that need to go in there. And the way that this input form works is that as you start typing, it will pick up on what you're writing. It will detect what the options are for that particular keyword that you typed, so you don't have to actually know what is the formal term for what you're writing in, right? It'll just help you pick what those terms are. And this is helpful. For example, here, you'll probably remember a lot more easily that your sample is human than it is 9606. Okay? But it does that automatically for you. Of course, if you're dealing with a different kind of sample or a species that's not listed in there, then you can enter a different term. And a good example for when that happens is, for example, if you're doing environmental proteomics or environmental mass spec, and pretty much the only thing you have is this is a piece of dirt that I collected near the river at this GPS location. Okay, so you can't really put in a species name in there, but you can definitely add some other information about what that might be. The next step is, if we really want to connect the dots behind the scenes, then if you want to say that this spectrum is this peptide, then we need to be able to connect those two things. And that's what this box here does, is if your results file says, I used this file name, in this MZ-IDENTML file, there's this spectrum file.mgf. Well, what's the name of the MGF in the spectrum files that I uploaded? What you'll often see is that the results files that your search engine generates might have a .raw at the end of your file name, but then you uploaded MZML, right? And those things don't match automatically, so then you have to make that connection. If the names are the same, then this is done automatically. You don't have to do anything manually. But if the names are different, then we need your help to connect those dots. Okay? But just in general, if the names are the same, if you didn't change your file names between searching and uploading, then this process just happens automatically. The associations, after they're done, they just show in here. It just says this uploaded file matches that searched file. Okay? And when all of them are done, then you get this green check mark, and that's the notification that you're good to go for a complete data set. So ideally, that's really what we would like to see for all of them. Final step, if you're submitting a proteomics data set, then you likely want to make a submission to Proteome Exchange, so you get a Proteome Exchange identifier. In some cases, like in metabolomics data sets, of which we have quite a few at MassIVE, then we recommend that it not be submitted to Proteome Exchange because it is Proteome Exchange. Okay. There's been some talk of metabolome exchange, but that doesn't exist yet. So as it is, that's really designed for proteomics data sets. If that's what you have, please do check that box. If you have a mix of metabolomics and proteomics, you might be better off making two separate submissions and having two different data sets. Okay? Or you can put them together, because as long as the data set contains proteomics data, then it's okay to submit to Proteome Exchange. And that's it. After that, you just click submit, and the data set submission process begins. So at that point, It will still take a bit of time to run because there's a lot of checks that are done on the data, but that's all done behind the scenes. It's not something you need to sit there and wait. It'll just do its thing, and whenever it's done, it uses the email that you put in your account and just emails you to let you know your data set submission is done. Okay? Are there questions about this process? It's kind of designed to be streamlined. I know if you're not actually doing it, it seems maybe there's less questions coming up, but essentially it is meant to be a streamlined process where the only part in there that might take a little bit more time is just making sure that the file names in your search results match the files that you upload. And that's really a check that's there to help make sure that things match. So when this is done, you'll see your dataset page will look something like this. You have an identifier for your submitted dataset. There's a title, there's a description. This is all the information that was entered in there, PI, submitting user, etc. And at the top, you'll have a few important tags. You'll have your proteome exchange identifier will show up there at the top. And if the data set is private, which it will be right after you submit, then you have that red label there that says private. Okay? And that means that only you, the person who submitted the data set, or anyone with the password can access it, but no one else can. Okay? And another bit of information that's useful to mention here is that data sets are immutable in the sense that once you submit it, we cannot change anything in it. We cannot delete files, we cannot rename files. And the reason for that is it's important to support the community in a way that if a paper is submitted for review and the reviewers, find something with the data that it cannot be changed. It is what it is. Now, what can be done is you can add files to a data set. So if you forgot something, the reviewers ask for more experiments, that's definitely okay. You can always add. We just cannot go back and delete it. However, if you really don't like it, just leave it there. Keep it private, no one will ever see it. No one will ever touch it. Make a new one that is how you want it to be. And then that new one can be used and the old one will just forever remain silent. Okay? Does that make sense?
So are you actually going to keep all of them forever? Like all of them?
That's currently the mandate that we have. The day might come where, if something is 10 years old, it's still private, the identifier never showed up in any publication, and we contact the authors and they say, oh yeah, that thing, there's really no reason for that, then it might go away. But as it is for now, the intention is that they will become available at some point, and for that reason, yes, the intention is to keep them around. But it might happen that would happen, and it would be more in cases, I'd imagine, where a substitute data set was submitted afterwards. That would be the case where it probably would make the most sense to get rid of the old one. Yes?
If you submitted the original submission to Video Exchange, you can upload the substitute data set or not?
But then upload what, I'm sorry?
Like a substitute data set.
Uh-huh.
Instead of, would you have to resubmit to Video Exchange or would you have to take back?
So what I mentioned applies to both sets of identifiers. So this example here, if this is a private data set, there's a private MSV identifier for the massive submission, and then there's a private proteome exchange identifier for the proteome exchange submission. And what would happen if you go to proteome exchange with that number is that, well, not this specific one because this is probably public now, but what it would do is it would show you a page that says this data set is private. And if you have the credentials to access it, then you need to go to the repository and access it. Otherwise, nothing else is shown. And that's actually a good segue into the next point, which is how do you give the reviewers access to the data set? Okay, and essentially what you need to do is after you get your submission in, you have this information here of the FTP link if they want to download the files, to look at the files in their, in their computer, or you can just give them the MSV identifier, and then they can access this page at MassIVE to see your private data set. But to be able to see anything, they're going to have to enter a username MSV_reviewer, and then you have to give them the password that you used when you created the data set. And only people with that password will be able to see anything. Okay? Otherwise, the only thing that anyone else will figure out is, oh, there's a data set with that number. But they won't see any information about it. Okay? So if it's a private submission, the only way to access it is to use the reviewer account and have the password that you created, which you would then have included in this paper that you submit. Okay. Typically what you see is that in that section of the paper where you talk about where the data was shared, you would just say the data set was deposited, it has the identifier, blah, 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 and the password for accessing it is this. Okay. Does that make sense? Has anyone used one of these as a reviewer? No? You might come across your desk one of these days. But yeah, so again, it's meant to be an easy process to be able to go look at it. Once everything is done, your paper goes through review, you're ready to make it public, you have a big button here at the top of the dataset page, and you can just click Make Public, and that's it. You make that click, the dataset goes live, it will appear in searches, it will become accessible without a password, and it will be announced through Proteome Exchange, so that anyone who subscribes to the Proteome Exchange announcements list will know this new data set has been released. And that doesn't mean it's the end. You can still do things with the data set after that. So if you at some point realized you were missing some files in your data set, you can go back and add. One example of when this is useful is if the original submission did not include metadata about which files go with which condition that you tested in your paper. And without that, it's hard to do anything about the biological conclusions of the paper. So it is one kind of information that tends to come afterwards. Or you can add metadata, as in maybe you didn't list one of the modifications that you searched. You can change the description, you can change the contact information, and you could add publications to a paper, to a data set, I'm sorry. It happens often that multiple publications result from the same data set. So as they come out, you can just add them in here easily. Okay, so that then people can find the papers through the data set in addition to finding the data set through the papers. Okay, and it kind of can work in a nice loop that way, that they start with one of your papers, they find the data, then through the data they find the other papers that also use the data, and so on and so forth. At least that's the intention here. All still good? All right, so, oh, where were we? This jumped to one. Okay, so for the next couple of things, this is an example of a data set. It's one of the probably most used data sets. It's a colorectal cancer data set from the Vanderbilt, and it's got 14,000 files, almost 1.5 terabytes of data. And it's one that we're gonna talk about quite a bit in terms of how to reuse data of the sorts that is shown in here. So I mentioned initially, and we can see again up here, there's 280 terabytes of data, 2 billion spectra. Now, does that mean knowledge? Have we, what have we learned from all of this data? And it's a bit of a sobering message. So let me take us through it step by step. So we started with looking at a subset of the data, focusing on human mass spec data, close to 1,000 data sets. And that's about 70 terabytes of data. Those 70 terabytes of data, 88,000 mass spec runs. A lot of instrument time going in there. So how much knowledge is there associated with all of this data? Well, the first thing that we realize is that 86% of the data is partial submissions. That means there's no reusable identifications. Maybe there's some IDs in there, but we have no way to parse them because they're in closed file formats. Okay, so we really can't easily extract that knowledge that might have been in there. 6% of the remaining, or the remaining 6% data format lost provenance. What does that mean? Like, you submitted protein IDs, but you never said what spectra that linked it to, or what spectra supported those protein IDs. So there's no way to go back and check, was the protein really there, or what peptide did you use to identify the protein? So that information is lost in those cases. 8% of everything, we do have result files with provenance. However, half of that has no false discovery rates. Again, what are you going to do with that? Some subset of that might be good, but we can't tell which subset of that. Or it reports an FDR larger than 1%. So the only part where we can have an FDR links back to the raw data and it's less than 1% FDR, there's only 1.7 million peptide spectrum matches. That's less than 1% of all the data that you could actually reuse something out of that. And I intentionally separate these two numbers because there's 14 million identifications that come from just one data set. But as you can see, that's not typical, right? It's 1% out of 70 terabytes, and then 14 million for just 1 terabyte. Okay? This data set was done that way because the NIH contracted it to be that way. So they were paying people to make good, reusable data. And that was, I'm not going to say it's the only reason it came out that way, but it definitely was a big incentive because it needed to be that way. So even if we include that, there's only 5% of the data that's minimally ready to be reused, but it's really less than 1% without that other data set. So it did bring up the question of how do we make this data more reusable? How do we actually derive some knowledge from this data? And this is where we really thought of MassIVE as, or wanted to think of MassIVE from the very beginning, as a different kind of repository, where it's not this typical view of repositories where essentially it's more like a data cemetery where data goes to die. That right before you publish your paper, you're forced to put the data somewhere and then you just, okay, where can I find a bin big enough to dump this in? And you go there, the data goes in, and that's it. Yes?
The same situation is right and other, or do they have different
You mean these numbers?
requirements? Yeah.
Oh, these numbers are proteome exchange.
Oh, project is.
This is across everyone. This analysis was maybe a year and a half ago, 2 years at most. And it was at that time using all the human, I believe, all human Q-exactive data sets that were available. So it was just across the board. There's a little bit more complete submissions since then because Mascot implemented saving results in NC-IdentifML. Before that, they didn't have it, so it was just much harder for people to make proper complete submissions. So things have improved a little bit since then, but not dramatically. So at this point, we're probably at 5% of the data could be reused, but it's still a very low number. Yes?
So if someone were to deposit his raw files and let's say MAS1 TXT outputs, would it be still partial submission?
Unfortunately, yes. there's been conversations with Jürgen over the years, and the thing is that I understand that Max1 is a piece of software that keeps evolving, and there's new things that they keep implementing, but in the process of doing that, they keep changing the file formats. And they're proprietary file formats in the sense they don't follow any particular standard. So the newer versions now are supposed to implement MCTAB exports, which would be a good step forward. So MassWeb would do MCIdentML, Max1 would do MCTAB. We support both. So if that begins to gain traction, that would definitely be a good way to move things forward. But in the past, it was not a complete submission because there were file formats that were specific only to Max1 and just kept changing.
So you define your knowledge as something that is automatically extractable.
Correct, because otherwise it's not something you can process across the board in a comparable manner, right? And then you can't really, I mean, essentially every single data set becomes something you have to invest a significant amount of time to extract that out. And we've done that to some extent with specific flagship data sets per se. So for example, the draft proteome papers in Nature, which is now about 5 years ago, right? Those were examples where the original submission was partial, but they had some identification files in there. But because it's such an important data set for the community, we actually went back and we converted all of that to open format and essentially upgraded the data set to complete by generating the open formats necessary to then access that information. But in the process, you know, some of the things weren't quite adding up, some of the things didn't quite match up, so I think in the end, maybe 90% of it was about the same. But it's really something that had the authors done it at the time, maybe that matching 90% would have been what they reported and not the remaining 10%. So, you know, it's better than not having extracted it, but it wouldn't be as good as them having done it up front. Does that address the question? Yes?
Yeah, what about the data like quality control? Like when you extract the data, how do you know there is a good quality or not?
That's a great question, and I wish we were close to being able to answer it properly. As it is today, pretty much data is deposited as is, and there's very little along the lines of doing quality control on that data. It is one of the major reasons why we have been getting involved with projects that use Max1 and OpenMS to process the data automatically for every data set. And we're going to talk a little bit more about that later in this lecture and then in the hands-on session also afterwards, where we will begin to automatically generate QCML or quality control ML reports for all the files that are submitted. Some of those quality control metrics are, for example, MS2 identification rates. But again, if you're making a partial submission, then there's no identifications at all, right? So that's a 0 right there. So from that perspective, there's little that can be done. So unfortunately, we're still at a point where the information required to derive proper QC metrics is not available, such as metadata, such as identification, such as those sorts of things that you can then use to do proper QC. So the hope is that by reprocessing, by providing ways to submit the data better, that we'll have the information to be able to generate those things. So as it is, we don't even have pretty much anything that we can reuse, let alone do the proper QC that should follow after that. So yeah, these are all good questions, and they are important questions, because if we want to reuse this data, we need to address these issues, right? So again, that's why at MassIVE we didn't want to think of it as just a way where you go to deposit the data. We knew that this was the situation, that unfortunately there's little information associated with the data, so there's something else that needed to happen to process it. And essentially, we think of it as an environment where you can go to reprocess the data and to extract more information from the data. So in particular, one of the features we have at Pride is that Honestly, we very much welcome your submissions, but we think of all the data that's publicly available as data that should be available to reprocess. So what that means is even if you deposit your data set at Pride, we will easily import it into MassIVE to be able to then reprocess it. As in, the first thing being just creating open formats for the files that you submitted. So we have ways to automatically convert from raw to MZML, and we have ways to convert your results files to MZTAP, which is an open format that then can make a submission complete. In the process, we also automatically reanalyze the data. If there's no identification submitted with the original data set, then the only way to get anything out of it is to reprocess it. And I'm going to talk a little bit about that with the reprocessing with the standard database search, but also with advanced algorithms that let you look for things that weren't there to begin with, such as look for modifications that were unexpected, or match it to genomics information to look for new splice isoforms, new exon matches, so on and so forth. And there's also visualization tools that you can use to interact with the data online. Okay, so let me give you a few examples of this. The way that this works is using this PROTEOSAFE system that we developed at UCSD. And what it does is, just skipping over most of the technical details, the point is just, we want it to be scalable. Whenever you launch a search in our system, it just runs in the back end in a compute cluster. You don't need to worry about that. It's going to run in parallel and be fast. That's pretty much what it is. It's flexible, means if you're a tool developer, it's easy to add a new workflow to the system. And because of that, we have over 60 different workflows that you can use to analyze your data. Some of them are for metabolomics, some of them are for proteomics, but it gives you a variety of different ways to analyze the data. As it is now, this system has been online for maybe about 10 years. So to date, we've searched close to 2,000 years of instrument time through this system. So there's been over 200,000 search jobs from over 45,000 users in a lot of different countries. And it essentially amounts to over 30 billion spectra run through the system to date. So just to give you an idea, it's not afraid of good data. Okay, so it's definitely ready to reprocess the kind of data that you get at the repository scale. So the first thing we did is, as I mentioned, there's so little information for the majority of these data sets, so we just reprocessed the whole thing. We started with all the human Q-exactive data that existed at the time when we started this analysis, and that was close to 40 terabytes, about 840 million MSMS spectra. And we reanalyzed all of that with the MSGF Plus database search tool, which is deployed in our system in PROTEOSAFE. And what we observed out of close to 20,000 spectrum files, we were able to derive over 400 million new identifications from this data. And this is closer to 30 times more than was available before in terms of identifications for data sets. Altogether, this is over 10 million versions, 10 million modified peptides identified in there. And all of these search results are attached to each data set. So what that means is that if you happened to have submitted a data set at the time that is human Q-exactive data, if you now go back to your data set page, you'll find a reanalysis in there that reprocessed your data and potentially found things in there that you hadn't seen at the time. Okay, that's now available for all of those data sets. And it's relatively easy to find if we just go to the MassIVE homepage. There's this box in here, the second panel from the top left, where you can search across these identifications. So if you want to look for all the data sets in which a protein has been identified, you can just go there, you get a query view just like that up there, and you can enter any protein name that you're interested in, or peptide, or modification, And then you can look for data sets, you can look for peptides identified for that protein, modifications, so on and so forth. Okay? And if you want to see what the evidence looks like, you can pull up the peptide spectrum matches and you can open it up and look at it. Okay? There's this Lorikeet viewer, very nice viewer developed by Vagisha at the University of Washington. We just reused it, integrated it into the system, and you can interact with the spectrum. You can zoom in, you can change ion labels, you can see the masses, and so on and so forth. So it really allows you to get an idea for how reliable are those calls that are being made in the system. Okay, does that make sense? So it now gives you a way to even begin to ask these questions, because it might seem strange, but without this, you couldn't even ask the simple question of what data sets has a protein been identified in? Or what peptides is this protein identified by? You know, those sorts of simple questions that we should be able to answer with all of this data that's available in the public domain. So now this begins to be a little easier to do. So how does this process work if we want to do reanalysis? The first thing is the reanalysis workflows that are available, the open source software usually wants open file formats for your spectrum. So the first thing that we wanted to do at MassIVE is make this a one-click thing. If you have a data set that only has raw files, click that button and just let it go. And a couple of days later, you'll have the MZML files in there, and you can search with any of the workflows. You can download it for offline analysis. You can do any other processing you want. But it's just a one-click button to go from raw files to open format MZML files. Once that's done, to initialize a reanalysis is also very simple. It's also just click a button that says, okay, I want to reanalyze the data in this data set. And that brings you to a data analysis site, our PROTEOSAFE system. And you just log on using your account. And the default workflow that is selected is just to do database search with MSGF+. And we're going to see later today in the hands-on session how you can also select, let's say, a MaxQuant workflow to do quantification analysis of the data that's in the system. Then the dataset files are pre-selected by default, but if you don't want to search everything at once, if you want to do a subset, you can just go in there and update that. There's a number of search parameters that you can set up in here. What enzyme was used, some of the modifications you might want to search for. And that's pretty much it. You just get a simple input form with the parameters for your reanalysis. And at the bottom of the form, you click Submit. Then it goes into the supercomputer center. You don't think about it. It runs in there. Some of the steps are parallelized. In this case, there were multiple raw files or multiple mass spec runs that were a part of the search. It just searches all of them in parallel, puts the results together, calculates FDR, does all of that. And when it's done, it just emails you a link to the results page. So you don't have to worry about how long it takes to run, is it done, is it not. Just forget about it until you get the link back that tells you this is done. And then at that point, you can see your search results, or you can download them, so you get them in open format, MZTab, which means you can use it to make a submission, or you can download it and use it internally for other purposes that you'd like to do. You can also interact with these views online. You can go and explore the peptide spectrum matches, you can look at the peptides, the proteins, so on and so forth. All of that information is automatically available in there. If you're happy with your results and you want to share it back to the community, it's also, again, a one-click from your search results to attach these reanalysis results back to the data set that was originally used to do the analysis. So if you click in there, you get a page that looks like a data set submission in the sense that you have to specify some metadata, but this is automatically filled up for you because it knows the data set you came from. Okay, so in this case, we started with one patient from the colorectal cancer data set. So it knows already that it's a human sample that was run on an Orbitrap. It had certain modifications. It just fills that out automatically. Then you ran a reanalysis, so it knows what files you searched. And it also knows what were the results of that search. Okay, so always through this process, we're trying to remove clicks if we can do it automatically for you. Okay, so you don't have to go back and find those files and put them in there. If we know where they are because we came from that place, we can just automatically fill in these options for you. The final thing that is shown in here is, this is something that you'll begin to see more and more as more data sets are reanalyzed, is that today when you submit a data set, you get a PXD identifier, which tells you it's your data set identifier. PXD stands for proteome exchange data set. But what you're going to start to see more and more is RPXD. What that means is a reprocessed proteome exchange data set. So for that, you just check this box over here, that when you do a reanalysis, it will automatically request that RPXD identifier for you, which you can then use for your publications. If you reprocessed an existing data set, that's the kind of identifier that you want to have. So those are pre-selected. You get the proteome exchange identifier. And final thing, you just, if your MZTAP file does not say what the false discovery rates were, if you ran it in our system, it will. You don't need to fill any of these fields. But if it doesn't, you can also specify it. And then finally, click submit. And that's that. That's all you need to do to attach the reanalysis back to the data set. And then what happens is that when you go to the data set, you now have a link from the data set to the reanalyses that are attached to that data set. And there can be different kinds of reanalysis. They have their own identifier. So in this case, there's the RMSV is the set of analysis for that data set, and .4 is one specific reanalysis in that data set, which would then have RPXD identifier that .4. Okay, the dots indicate specific reanalyses within a data set, of which there might be several. Okay. Does that make sense? Okay, and for all of these, once you open the reanalysis, you can see all of the MZTAP files. And really important, it's something I really like and really want to emphasize going forward, is that to the extent possible, we be able to see the full provenance of the results. What do I mean by that? It's not just what identifications you got, but it's how you got them. What search engine did you use? What search parameters did you use? What database did you use? So for these searches that are run in our system, clicking this button, View Source Task, takes you back to the exact search that was done to get those results. And all the parameters are in there, all the information's in there, and you can run it again. You can change things a little bit, run it again. Or you can take that same search protocol and now apply it to your data. And ask the question, if I do the exact same search, am I going to get results that are comparable to theirs? Okay, so this kind of provenance is very important, because otherwise it's hard to know exactly how the results were derived. But at least, first, that there are recent results, and second, that we'd be able to find the provenance. So when you do click to reanalyze SPECTRA, I just described the process of what happens when we run an MSGF plus search, but there's a variety of other tools that are just up front, the types of things that you might want to do. So there's a SPECTRA library search tool in there also, MSplit. There's proteogenomics searches that look for new splice isoforms, new exons, those sorts of things. That would be the NOC workflow. There's a blind modification search engine. We use MAESTRO. There's MAESTRO, which I'll discuss in a little bit, that actually puts all of these three things together, where actually MSplit, MSGF, and MAESTRO, and does them all one after another. I talk about that in a little bit. And then there's also spectral library search for swath data, MSGF DIA, that we developed at the time. And there's a bunch of others, but just to give you an idea that there's a variety of different ways in which you can reanalyze the data. It's not just MSGF plus. And what that does is that over time, you see the same data set gets analyzed with different tools in different ways. And when you run it, when you run these analyses in different ways, you get different results from each of them because you're learning different things from each of them. So for example, with MSplit, you might find mixture spectra. With BOD-A, you'll find new modifications, things that you hadn't considered in your original search. So to be able to get a sense for what those things might be, you have also a results comparison workflow in the system that allows you to take this search and that search and compare the two. And ask the question, well, what about these two is in common versus what is unique to A, what is unique to B? And you can look at that question in terms of the data sets that are in the system. You can select reanalysis of data sets or results from data sets, or you can select results from your own searches of your own data. So the system allows you to run searches on the public data, but also on your private data. Okay? As a rule of thumb, anything that you upload to the system is private. It only becomes public after you go through a series of steps to make it public. Okay? And that's how we like to keep it. That first and foremost, your data is yours, and it only becomes public if you say so, very explicitly. Okay? So you can definitely have your private search results that you can use, you compare against public results, or you can compare two of your own searches also. That's, of course, also fine. And then you get these types of views. You can see who gets more identified spectra, or you can look at it at the peptide level, protein level, so on and so forth. This remapped protein level, it's there because sometimes the searches are done using different databases. Like someone might search RefSeq, someone might search UniProt, and then the protein results would look totally off just because it's different identifiers, right? So just to get rid of that problem, remapped proteins just takes the peptides and maps them back to the same database. Yes?
You said proteins, and that caught my interest because what tricks or gimmicks do you do for protein interference? Because my impression, for example, at one point was that this depends to an astonishing amount on the actual tool you use for protein interference. And then when I used MSGF plus in front as a search engine, I got again different results on the protein level, although the peptide might have looked similar.
Right.
And I'm really intrigued.
Exactly because of that, we primarily do the comparison at the peptide level.
Yeah.
To not try to make too many calls on that front.
Yeah.
So I guess the first answer to that is the proper, where we can make a more proper comparison is for proteins that are identified with unique peptides.
Yeah.
Right? In that case, there's no ambiguity as to where it comes from, and that's what it is. Now, other than that, essentially what the different approaches do is take different forms of maximum parsimony. And it's try to pick the protein that explains most of the shared things and select that, take that from the set, go on to the next one. What varies between the approaches is the scores that they use, right? And that's why you sometimes will get different results there. So it's not something we can solve in this comparison. What we can do is just tell you, look, these are the ones that appear in both versus those that don't.
But do you have a tool for protein interference? You have MSGF plus, and whatever, but is that something you actively work on?
So not specifically in the sense that there's a separate tool for that.
Yeah.
We did have a paper, I don't remember how long ago it was, but essentially what it does is to take the approach that the evidence you have, or the score you have for a protein, is the accumulation of independent evidence from all the peptides of that protein. So you just score the proteins based on that, and then we do the same maximum parsimony selection based on that some of the spores for the protein. So it wasn't in and of itself a separate tool,
Yeah.
but just something that's incorporated into the workflows. Does that make sense?
It makes sense. Can I ask another question?
Sure,
Am
sure.
I going on everybody's nerves? We played with Fido protein infusions
Okay.
for a while, and
Uh-huh.
someday we were happy, someday not so. Any comment on that tool?
I haven't used it,
Yeah,
so I can't tell.
the developer disappeared a bit, so that's why I'm also a bit worried.
Yeah, that does tend to happen, unfortunately. No, I think we'll have to chat a bit more about it afterwards, but I don't have direct experience with it. All right, so we do the comparisons. What do we learn? Why do we even run separate analysis? And this is why we do it. So for example, in this particular case, looking back at this colorectal cancer data set, the original set of results had close to 7 million identified spectra. The MSGF plus reanalysis gave us about close to 9 million identified spectra. Spectra. What else did you learn? So if you do the spectral library search, we actually identify a lot of mixture spectra. So that's an example of something that the previous searches could not identify. And those are spectra where essentially you have more than one peptide in the spectrum. That happens quite a lot. And it's just most search engines just make the assumption that that doesn't happen. They kind of close their eyes to it, but it does happen quite a bit. So that's one thing that this workflow can inform that the others mostly don't. Proteogenomic searches, looking for different kinds of genomic events that are not represented in the original database, identified about 20,000 proteogenomic events, as in most of the time it's new splice junctions that were not in the original database. Some other times exon boundaries were corrected. There's some out of frame identifications. So there's other kinds of biological events that can be derived and identified with that kind of analysis. And then blind modification search found a lot of different modifications in the system. So all of these are probably things that are useful to learn about a data set. And it's good to be able to run these different searches. If you're interested just in one thing out of these, you can go just directly into that and run that kind of search and see what the results are. But honestly, we all have busy lives, and generally speaking, you don't want to go and do a proteogenomics identification before you first looked in a spectral library to see if the spectrum is already identified. Or you don't want to look for a more surprising explanation of the data before trying the simpler things first. So I kind of tend to prefer the approach where you can just click a button and things happen, okay, and does all of it behind the scenes. So that's why we developed this MAESTRO workflow. And what this does is the following. First, it takes all of the data and just clusters repeated spectra from the same peptide. If you have, a bunch of samples, a bunch of patients, if it's the same spectrum, it should be the same ID, just put it all together. Then, first thing after that is to do a library search. If we've already identified that spectrum in the past, we're pretty sure what that spectrum is, let's not do that process again. It just matches to the library. If there's an ID, okay, we're good. If there's no ID, then we pass it to database search. Okay? If it's not yet in the library, okay, then let's look in the database and see if we can find a new peptide that hasn't been identified before. If we have, we move it down. If not, then only after these two stages, we go and look for unexpected modifications. Okay? If it's not in a library, if it's not in a database with your standard modifications, okay, then maybe it is an unexpected modification. And these modification searches are also searched only against proteins identified from the first two stages. This does make a trade-off. If your protein is there only with an unexpected modification, it will be missed. But how often is that the case? And how often is the case that you want to find a new modification without having any other evidence for the protein. So most of the times when you have that, those are the false positives. Okay, so that's why we take this approach, with the understanding, that in some cases it might indeed happen that you might lose an ID because it's only there with an unexpected modification. But the general situation is that that's not the case. So all of these are then put together with variant level FDR. What this means is that the modifications are taken into account properly when calculating the false discovery rate. And it's a single false discovery rate for all of them, so that it's balanced across all of those. And then it combines with some quantification information. Initially, this is done with spectral counts, but we'll move it on into more intensity-based quantifications, and then you can visualize the different related versions of the same peptide in this network type of view, where each node corresponds to a peptide, and there's an edge between two nodes if the spectra of the peptides align significantly. So if you have two repeated spectra of the same peptide, they're naturally going to match. But let's say you have the peptide and the oxidized version of the peptide. Then the peaks will either match exactly at the same position if they don't contain the oxygen, or they'll be shifted by 16 if they do contain the oxidation. So what the algorithm does is it detects those mass shifts and still matches the spectra. It looks at the parent mass difference. Oh, you differ by 16. Then it looks for peaks that match exactly or shifted by 16. And if that is a significant match, then we get an edge in this view. And that's why it's able to then display all the information about a peptide in this way, where you can see all the different versions. Well, you probably can't see because it's too small here, but when you have it on the screen, when you pull it up, you can see how much of each is in each condition. Here, blue and red are cases and controls. And you can see how they relate to each other in terms of the modifications that are on them. To give an idea of what this allows us to learn from the data that we have, here's an example that takes into account 3 different data sets. And they're all 3 different colorectal tissues. And 2 of them are from the draft proteomes that came out in Nature a few years ago, from the Pandy group and from the Kuster group. And then the 3rd one is from the Matthias Mons group, which is a more recent data set, but it also takes into account more data. And it's on a more recent instrument, so you actually get quite a bit more spectra. Well, actually, no, it's less spectra than the other 2. But this is only colorectal. That's the total number of spectra for the 2 draft proteomes. So running these 3 data sets together through the MAESTRO workflow I just described, we're looking at close to 10 million spectra identified. This is close to 70% identification rate for those data sets. Pretty good. And we get about 160,000 unique modified peptide identifications in here. And 63,000 of them have some modification on them. Okay, so that's about close to 20% of all the spectra, close to 2 million spectra. Now this is something that really surprised me in this data, and continues to surprise me in general when we do reanalysis of data, is that I expected that a data set would have 20 modifications, maybe. But actually what we find is over 100 modifications. And by this I don't mean 100 different modified peptides. I mean 100 different types of modifications. As you go to Unimod, you'll get like 2 lists. I believe they have over 1,000 in there now. And we can see many, many, many different types of modifications in most data sets. In this case, in this analysis, there's 137 modifications with 10 or more peptides. And then there's 85 rare modifications that occur in less than 10 peptides. Are they true or are they false positives? Well, if you look at the p-value of the alignment of the spectra between modified and unmodified, it's very low p-values. very strong p-values, right? This is a log 10 scale, minus log 10 scale. So this is 10 to the minus 20 chance of that being a false match between modified and unmodified. So very strong evidence that those are true identifications. Some of them with very small number of identifications for that modification. Now I would argue that these would actually be the ones that are most likely to be biological. Because if you have a modification that happens on 1,000 peptides, it's more likely a sample handling modification. Unless you did something to your sample, like if you enrich for phosphorylation, sure, you're going to see it all over the place. But if you didn't, and you find it, that's probably significant, right? But on the other hand, if you find something on 10,000 peptides, you're probably causing it yourself. If biology was doing that, then it would be pretty much out of control, happening all over the place, right? So you want it to be a rare event. So what we found here, there's also a lot of putatively novel modifications. This is after excluding possible combinations of modifications, possible polymorphisms and other things. And in particular, this one amino acid in albumin, cysteine position 289, exists or is detected with 24 different modifications, 24 different types of modifications just on that cysteine. And is covered by 163 different peptide variants, as in a peptide with different combinations of modifications, covering just that one site. So this just gives you a sense for how much diversity is there in a data that's currently hidden under the hood. And it's likely also missing in your own data, right? We're finding this as we reanalyze the public data after it was shared, but the same thing would happen in data that is not shared, obviously. It's just either it's not being looked at. So looking back in a network view, we can see here for the different data sets and the different cohorts in the Matthias-Mann data set, we have controls, adenoma, and cancer. So there's 3 different groups in that data set, and the other 2 data sets, mostly it's just one group. And you can see how much of each peptide is identified in each of them. The size of the nodes is just a log scale for the peptide abundance. And then the pie charts show the breakdown between the different data sets. For this particular peptide, there's all these different modifications that are detected on it. And you can see some of these are very specific to the Matthias-Mann data set. And that's not surprising because this is a data set that was stored in FFPE, and that in and of itself causes modifications to the sample. Okay? So of course, all of those modifications that are caused by the formalin fixing are then detected here, and of course are much more abundant in that data set because that's where they should be happening. So what we're finding is this general sense of hypervariable regions. There, if you look at the full data set, all the peptides identified in your data set, the average number of modified versions per peptide is very low. It's about 1.5. This is good. If I didn't see this number, I'd be very worried. Okay? You shouldn't see an average of 10 versions of every peptide. Okay? That's not what you should see. 1.5 means about 50% of the peptides get an oxidation or a pyroglue or something like that. So this is not something that happens all over the place across the board. But there are some peptides that are hypervariable. There are some regions of the proteins that have lots of modifications. And this is an example of one region that has all of those modifications shown in there. So just quickly at the protein level, it looks something like this, where at the top there's a protein sequence shown here, then right above is the coverage in terms of speckle counts as they're identified, and then underneath there's a representation of the modifications detected at the amino acid in that position of the protein. So there's different types of modifications shown here: artifacts, chemical derivatives, post-translationals, glycosylation, amino acid substitutions, so on and so forth. Many different versions that can happen in there. And if you see the same symbol multiple times, it means different modifications of that type were found at that location. So the point here, obviously, not to emphasize too much which modifications detected where, but mainly two things. First, a lot of them. Okay, a lot of modifications, and not just artifacts, but many post-translational, many chemical, many others that are enzymatic that you'd expect to potentially have some meaning in terms of function. And the other point being, not really that correlated with expression or detectability of a particular peptide. We see them all over the place, pretty much regardless of whether we see a lot of peptides for that region or not a lot of peptides for that region. Okay? It just seems to be more correlated to what the protein actually is than the specifics of the sequencer of the coverage. Okay. Does that make sense? I wanted to see where we are. This is the other option. Any questions on this part? The modifications that have come in there? Okay, so I'm gonna skip, yeah, go ahead.
When you do this blind modification search,
Right.
do you search for, do you have a list of modifications that you go through, or is it kind of an open search and you have a delta mass?
Very good question. So we do the search itself, we do with delta mass. We allow for any delta mass to occur anywhere. And it is known and expected that obviously that gives an advantage to decoys. So then what do we do after that? We have a separate step we call mod decode, and we hope to submit that soon. But essentially what it does is it takes all the delta mass IPs and does two main things. First, do we have the unmodified peptide? And if we do, does the fragmentation match what we're seeing? If it does, that's a huge confidence boost that this is actually something real. Now, I will also acknowledge that there are modifications that change fragmentation, and that might reduce that similarity. We do some things to deal with that. But the point being is, if it matches, we really believe in it more. That's number one. Number two, what we do after that is now we take all the modifications that are known from Unimod, and we try to fit to that delta mass. First, we start with artifacts, and we start with frequency, right? So the first thing you're going to see is just oxidation on methionine just takes a lot of those items. Then you find all the common pyroglues, all the common modification stuff. After that's out of the way, we begin to look for, okay, what else might explain those deltas? And that's where you start to see the other modifications that are actually more useful. Okay? They have to match by mass, and they have to match by site, and there has to be no runner-up explanation that makes sense. As in, if I combine two oxidations plus a pyroglue, what does that look like? So we also look for combinations of modifications and for polymorphisms and all of that. And only after all of that's out of the way, we give you the possibility, maybe it's a novel modification. Okay, so first it tries all the possible explanations given what is known. And only if all of that fails, it goes into putative analysis.
Thank you.
But all of them also have to have this really good match between modified and unmodified. And that, I feel, is something that's very important for modifications analysis, because if we just give you a list of delta masses, might as well not give you anything, because it's really hard to process that. You don't know which one to work with, right? Because you have, what did I say, the 2 million spectra with these things? Which one to start with, right? This at least gives you a starting point for where to look at. And it does resolve them usually very nicely because of this correlated fragmentation. All right, so I don't want to not address questions, but I also don't want to not get to the quant part. So let me skip a little bit and go forward to slide 44. So skipping a couple of things that were in there, because I want to go back to this question, does data equal knowledge? And all that we mentioned about here in terms of ID, that is all this breakdown, well, 0% of data sets have quantitative information. Okay? Nothing at all. There's no information, there's no complete, there's no partial, there's no open format, there's no closed format, it's just pretty much nothing. I mean, if you want to throw it a bone, you might want to say, well, the spectral counts if you have IDs, but that's about it. And you know, that's pretty poor. So because we're very aware of this dire state of affairs, we've partnered here with Mina and Olga, and we're putting together this effort that we call MassIVE Quant. And the intention is that we begin to have a way to represent quantitative information for data sets, and to show not just how you can represent that information, but also keep information about how the analysis was run, so that you can go back and reproducibly get the same results. Okay? And that involves keeping track of the intermediate files, involves keeping track of software versions, involves keeping track of a number of things that have to be written into the platform to make it easy and accessible to use this kind of information. So I'm going to give you a bit of an idea of where we are with this at this stage, and then when we get to the hands-on session, you're actually going to see how you can already use some of those resources to run your own analysis and also submit quantitative information. So the first thing that is important for this is when you submit a data set or a reanalysis, you can add quantification results, where that would include annotation files where you specify the metadata. It would also include your MS stats input and output files. And then in methods and protocols is where we recommend that you submit your scripts and other files used to run the analysis. And you're also welcome to deposit there, let's say, a zip file of the of the version of the software that you use to do the analysis. All of that is supported and can go in there. To describe some of these features, I'm going to use one of the data sets that is available in the system already with quantitative reanalysis. And this is the data set from an IBRF study that was run in 2015 to look exactly at questions of quantification. There was some older ones, so it was an outdated type of study, and it's great that we have this new one available. And so here is shown one of the plots from the paper in terms of the results that were submitted by the different participants. So each of these columns is a different participant in the study. And we see here that below this line is the number of true positives. This dashed red line is the one that you want to touch underneath. On this side, it's not the good direction. This is the number of false positives that were reported by those studies. Some of these just seems like they didn't quite apply a threshold. They just reported everything and forgot to distinguish between what's differentially expressed or not. But really, what you want to know is what happens down here, right? If someone submitted that kind of result, what pipeline did they run? How did they run the analysis? How can I go and do the same thing on my data, right? How can you learn what happened in there to do the same things? And that's the kind of information we want to capture, is how could we go back and, let's say, pick this one or pick that one that is so close to getting pretty much all the true proteins and yet no false positives, right? So that's the situation we wanted to be in. So this is in the system right now, and it's this data set here. If you click up there on that link, it will take you to the repository, and you can see this data set in there. And there's 29 files, close to 18 gigs of data. And now there's some reanalysis that are already in there. So if you open this data set page and go to the bottom there, you'll see there's already a number of reanalyses attached to this data set. There are some MAESTRO search results. Then after that, there are 3 reanalyses that Mina submitted that have the details of how you can do a skyline reanalysis of this data, how you can do a max quant reanalysis of this data, or a progenesis reanalysis of this data. And these were done offline by the tool developers to run it as well as it could be run according to how they designed the software to run. Okay, and now you can go there and see what are the details of how the software was run. The last three that are shown in here are actually reanalyses that you can now do directly at MassIVE, at least for the part of running MaxQuant and running the search tool. MSStats is in the process of going in. We'll get it there. We'll get it there. For now, it's still offline, but it's also a part of that reanalysis. And we'll see in the second part today how that process actually happens. We're actually going to run through one of these. We won't have time to wait for things to finish, but we'll at least be able to get it started. So when you click on one of those, let's just say we click on this MaxQuant reanalysis, then you get to see what the reanalysis looks like. And in particular, you have this really cool new button, which is quantification results. You can actually see some quantification results from a reanalysis. And then you will see things that should be somewhat familiar to you at this point. You see the annotation file that is given to MSStats. You see the evidence file and the protein groups that are also passed to MSStats for the analysis. And then after that, we have the results as were derived by the analysis of the files passed in there. Okay, and if we went to the reanalysis and looked at the scripts part, or the methods and protocols, you'd actually find the actual R script that was used to run MSStats. So you could reproduce exactly the same output by using the same inputs that are there in the system. Another thing that's probably also familiar to you at this point is what the output of MSStats looks like. And what the system does is just parse that automatically and just show it to you as a table that you can then filter and sort. And most likely what you'd like to do is filter your adjusted p-value by your significance, your desired significance. Select only cases for which there are no issues. And then if you want, you can sort by any particular column also, in this case, sorting by full change. Or you can download the results for offline processing or for whatever else you might want to do. You'd essentially get pretty much the same table. If you download, you get pretty much the same table that you would get if you run MSStats offline. Okay. So now becoming able to actually express this in the system, to actually see this in the system, and at the same time have information about the metadata that is used to derive this. What were the scripts? Okay, so all available in there. So as it is today, we can run the analysis in these three different types of workflow. We can run MaxQuant with Andromeda in the system directly. Then you can download and do the MSStats analysis offline and make it into a reanalysis. There's one in there right now showing this direction. Or we can do MSGF plus search, do the MaxQuant for the feature extraction and intensity integration, combine the two, run MSStats, it's also available in there, and the same pipeline but using MAESTRO searches. Essentially what we get after that is pretty much comparable results. There's a couple of things that change here and there just because of one more peptide, one less peptide, bringing a protein in or kicking a protein out, so things kind of vary just a little bit between the three different ways of running the search, but generally speaking, pretty comparable. And actually, I should correct this in here. When I say 25 proteins, 26 proteins, what this really means is protein condition pairs. There aren't 26 proteins in there, there's only 6. But you're comparing condition 1 with condition 2, 1 with 3, 1 with 4. So this is counting the number of those pairs where proteins were found to be differentially expressed. So this should say pairs and not proteins. But the main point here, just through the volcano plots to give you an idea in terms of log2 full change, log 10 p-value, that the results are pretty comparable, especially when you get out from this part down here, where it's the less significant IDs in the lower fold changes. As you go out from the more significant and more pronounced changes, the results are pretty much comparable with all of So I wanted to make sure to get to this. And also to acknowledge the people who did pretty much all of this work. Jeremy and Ben, very instrumental in developing all of MassIVE, all of these features that we use at MassIVE. Julie was the main developer of the MAESTRO workflow, the reanalysis that I mentioned, that we reanalyze those 40 terabytes of human data. Xunjin for MOD-A and modifications analysis. We talked a little bit about how the modifications are decoded. That's also Xunjin's work. And of course, Mina and Olga for the massive one part that we've been now developing, and I'm glad that we're able to have and run it in just a little bit when we come back to the hands-on session. So with that, we're pretty much at 10:30, so we'll wrap it up for this session, but love to hear any questions you might have or any comments. Feel free to come up. Other than that, I guess we know.
Break until
Break.
11.
All right.